Couple of quick announcements before we get started. Blatant plug for the lab that I'm teaching next term, Mutant Viruses from Hell. I think today's the first day you can sign up for it. Got any questions, come find me after class or send me an email. Another, I think, should be really interesting presentation will be today at 11 in room 409 in SB1. This is a discussion by one of the master's students in Justin Corcell's lab who's going to talk about transcription. I got transcription on the brain. Replication in E. coli, and particularly what happens at termination. So we talked a lot about origins of replication and how those work and the various different proteins that are involved. Termination is actually a lot less well understood. And so that's one of the things that the Corcell lab has been working on. So that's, I have no idea if any of that stuff is going to be on the exam. Shouldn't be, because only stuff on the exam will be what we've covered since the last midterm plus today. So whatever we get through today will be on the exam on Friday. No new material on Wednesday. So we shall see. Anyone who's looked at the syllabus recently may have thought at the beginning, oh, well, where the heck is he? He's like ridiculously behind. But actually, we're caught up as of today. So syllabus should be more or less correct as far as that's concerned. A couple people have asked me about lecture recordings. I literally sent them a nasty email yesterday. They wrote back to me. We shall see. Hopefully all the recordings will end up working. I did post an audio, thank you for the student who provided that, for the last lecture that did not get recorded. There's a little gap in the one from Friday, but hopefully most of that will be there. Okay, any more questions about exam, things coming up? Those of you who are taking the exam in the testing center, please remind me so I make sure they get the exam over there. If you're not, you could also let me know, but that's not as critical. More exam question things. Okay, so let's get started with our presentation for today. What did I promise? Oops, at the beginning last time? Clicker question, exactly. Nice review. So by the time I get myself fired up here, I'll have a chance to get your clickers out. The presence of which of the following factors in a specific cell is thought to determine whether or not an exon is used for alternative splicing? BBP, SR proteins, the 5' splice donor, the 3' splice donor, U4, U5, U6. And we're still aiming for 100%, right? Five. Okay, we're not at 100% by any real stretch of the imagination. So let's stop and chat amongst yourselves about why you answered what you answered and why you might want to think about changing your answer or not, as the case may be. Ready to go again? Yes, no, go. 10, 5, okay, stop it before I show you what people answered. Always a good idea. So, presence of which of the following factors in a specific cell is thought to determine whether or not an exon is used for alternative splicing? U4, U5, U6 are going to be present in every single different cell. It's the difference in different cells determining the different splicing are going to be these SR proteins. So that's the answer. And probably, I was just wondering myself, maybe we should just do this for the exam so everybody can chat between themselves during the exam? Yes. Sorry, not this year. Maybe next year, when I'm on sabbatical and won't actually be teaching. Okay, so wanted to spend a little while longer talking about introns. Again, one could spend all term talking about introns. A couple of things that I actually find to be really fascinating, and particularly as looking through the lecture from last time, I showed you all of these amazing images of high-resolution structures of the spliceosome and all these extra proteins and RNAs and all of this really ridiculously complicated aspect of what's going on with splicing. But it turns out all you need is the intron. You don't actually need any of these proteins. You don't need a whole bunch of different RNAs. You just need one RNA. And so that gets me to the whole concept of what people also call ribozymes. And this is actually kind of a fun story because Tom Check, who was working on this, I interviewed with him for graduate school actually right before he got his Nobel Prize. And he was telling me this story about how his students came and told him, because they were trying to work on splicing, and they said, we were trying to do this control. You know, we had to mix all these proteins together, we got splicing to work, and then we left out all the proteins, and the splicing still worked. And then he said, go back, do it again. They did it again. He still didn't believe them, went back and did it again. And after about fourth or fifth time, apparently he finally believed them, and then he ends up getting the Nobel Prize for this. So what they had discovered was something called self-splicing introns. And it was actually they called group 1, because these were the first ones that they found, set of self-splicing introns. So it was literally a piece of RNA that would cut a smaller piece of RNA in the middle of it out and hook those two ends together, just by itself. Completely purified RNA, nothing else which was in there. And so this was very surprising, and again, nobody really believed it, but eventually it was shown that it was literally the RNA which had this enzymatic activity, and as we talked about U6 last time, it does seem to be the U6 part of the spliceosome which also has the enzymatic activity for doing the splicing. There's one however really, really big difference with these self-splicing introns versus what we now know as the spliceosome. These are all completely dependent on the sequence in the intron. So again, not surprisingly, you get all the specificity from RNA-RNA interactions, again, base pairing interactions that happen with each other. But if you have exactly the right sequences in your intron, then that intron can cut itself out. But that's not terribly useful if you're going to use introns in all kinds of different genes for alternative splicing. You need SR proteins, et cetera. So probably a very inefficient way of doing splicing in general. And it turns out that there are two different kinds of these self-splicing introns, again, group 1 and group 2. It really does seem to be these group 2-like introns. Now, they're feet and hands because the authors of the textbook thought they were being funny. But their big difference here is that in group 2 self-splicing introns, there's this adenine where you have the 2' 5' OH bond that happens. And in these group 1s, actually, you need an external guanosine, which is actually not part of the sequence, which makes this link here. But otherwise, the cutting, rejoining, et cetera, are all really pretty similar to each other. And this is, to me, kind of an amazing example of evolution. It's probably there was some kind of self-splicing intron that developed, again, evolutionarily speaking, and then this mechanism and specifically these sequences. So again, this is all the sequences you need for splicing are here in this self-splicing intron. All of these got farmed out to the rest of the spliceosomal components, and evolutionarily speaking, it turned out to be useful to have these introns and what those introns would be for. Now, we talked a little bit last time about alternative splicing being a wonderful thing and being able to mix and match different kinds of exons to give you slightly different proteins. And this happens in very, very many of our genes. But as one of your colleagues was asking me about before lecture, this seems like a really inefficient way to go about making your genetic material. You make these really long primary transcripts which have exons and introns in them, and the exons are really short and the introns are really long. You're just throwing away all of those nucleotides. Well, throwing them away is maybe a little extreme, but one of the things that people have been looking at recently, and when I say recently, I mean as recently as last month, have been maybe there are some other roles for introns in cells other than giving you the opportunity to have this kind of recombination taking place. So to talk about this, I need to give you a little bit of background on Saccharomyces cerevisiae. When we talk about eukaryotes, particularly in terms of this course, 90% of the time, maybe just 80% of the time, when we talk about eukaryotes, we're talking about Saccharomyces cerevisiae because these are really easy organisms to work with. You can make very large numbers of them, basically kind of treat them like bacteria. There are no yeast rights groups as far as I know, so you can kill billions of them and nobody really cares. And probably the most important is that there's really good genetics for yeast. And so you can make lots of different kinds of modifications and still work with these organisms. A couple of differences, however, between us and yeast, and that is that only about 5% of the Saccharomyces cerevisiae genes actually have introns. So pretty small compared to the 90+% that we have with introns. The other thing that's different about the introns in yeast is only 9 of about the 5% of genes in Saccharomyces cerevisiae have more than one intron, whereas our introns, we have an average of 8 introns in any of our given genes. So that means that if you want to study what introns are potentially important for, you might want to study in a really simple system that has very few of them. And so you can actually start to do some of these experiments. And so these are the 2 experiments I'm going to talk about. We're going to look at some of the data for both of these papers. Parenteau, which I think is how this person pronounces it, published again last month in Nature. What they did is they went through the almost 300 introns in Saccharomyces cerevisiae and just knocked them all out. Again, great genetic tools. So you can literally go in and take out every single one, again about 300 introns, in the yeast genome. And they found about 5 of them were essential, which actually is kind of surprising. You know, only 5 of them of the 300, and very, very few that you actually have. So that was interesting, and they actually were kind of surprised. But then what they did is they did some really neat experiments that we'll take a look at in just a second, is they compared yeast that had introns to those that didn't have introns in the stationary phase of growth. And so stationary phase, which you probably should know. How many of you had micro? How many are planning on taking micro next term? Okay, so stationary phase is after you have logarithmic growth. So logarithmic growth is over a certain period of time. In the case of E. coli, it's every 40 minutes. You go from 2 to 4 to 8 to 16 cells. But if that happened over any reasonable period of time, there wouldn't be any resources left. We would have completely consumed everything, and this room would be filled with your bacteria, as the case may be if you're just talking about E. coli. So at some point, you run out of nutrients. And so that's what happens in what they call the stationary phase. Same thing again happens with yeast. As I mentioned, yeast are a lot like growing bacteria. You can grow them for a certain period of time. They start to run out of nutrients. And then they go into this sort of resting phase. And that resting phase is probably what most organisms are dealing with most of the time. Hopefully all of you had breakfast. That would be good. But if you're starving, you're not quite as up for, have good breakfast before exam on Friday. So basic idea here is you're not really happy, but if you get some more food, you'll be happy again. So hypoglycemia is another way of thinking about it. So what was interesting is a bunch of these intron-lacking Saccharomyces cerevisiae strains had a real problem in stationary phase. They were not happy, and they were less happy than the wild type. Then probably even more important, whenever you see something like this, you do an experiment, you get rid of a gene, and you see something that happens, you see a phenotype, can you rescue that phenotype? And so rescuing the phenotype is literally by putting whatever you got rid of back in and showing that you get back what you started with. And we'll see that's also called complementation. So it turns out that's exactly what happened. And then at the same time, another paper was published. Now, whether this paper was actually happening the same time independently, who knows? But at the very least, another paper basically was looking again also at yeast, also at RNAs, but now instead of getting rid of introns, they were just looking at sequences which were accumulating in the stationary phase. And it turns out, very strangely, introns were being accumulated in the stationary phase. But introns were supposed to get chopped up and gotten rid of. So what the heck were they doing left around in the stationary phase? So let's look at some of their data. This is from that first paper, the paper where they knocked out all of these individual introns and then just looked to see what happened. And so these are graphs which it took me a while to figure out exactly what was going on with them. So what we have here is relative amount of a particular messenger RNA. So these are the messenger RNAs that are up here at the top. So how do you know the relative amount of a messenger RNA? Use a technique called RT-PCR. Now, we haven't really talked about RT-PCR, but what's RT stand for? Reverse transcriptase. So reverse transcriptase followed by PCR. We've talked about PCR. So this is a way for looking at amounts of messenger RNAs. So you make a DNA copy, and then you look at how much of that RNA you had after making it into DNA. So it's just a way of quantifying the amount of one specific RNA. And then what they did is they compared the wild type, which is in the dark gray, to the gene when it's lacking the intron, delta I. And what they saw was in the logarithmic phase, the relative abundance of this deleted version versus the wild type of version, pretty close to 1. So no big difference. On the other hand, in stationary phase, they found that there was way more of this wild type than there was of the deleted strain. Same is true of this YSF3. DYN2 is interesting. Again, these are just names of different yeast genes, and they seem to be pretty random, relatively speaking. Is pretty close to nothing, but actually the wild type and missing the intron were very low in stationary phase, and the same thing was true with this other gene as well. So these genes are much, much, much less expressed in stationary phase when they're lacking these introns. Okay, what else is going on just to the cells? And so this is that whole idea of stationary phase. This is a growth curve where you look at just the absorbance of your culture, which correlates to the number of cells. It's looking just at the optical density, which you can correlate with cells. And what you can see here is that if you just look at the black line, this is the wild type yeast, it grows, and then at some point it stops growing and kind of flattens out here. If you now take the yeast which is lacking in the intron in this gene, the red line is lower and happens a little bit earlier than this one. If you put back the intron-containing gene, the blue line is back up to this line. And so what happens here, okay, so we've got these lack of expression in stationary phase when we do this deletion. And stationary phase happens earlier and you get less growth in the absence of an intron, but you get it back when you get your intron. So there's something about the presence of these introns which helps the cells to survive in stationary phase. So this brings us to the next experiment, which is basically doing a very similar thing, where if we just look at, and this is a nice review for splicing, regular spliceosomal splicing. Here you have 2 exons and your intron with the G with the 5' phosphate. It's going to attach to the 2' OH in the branch site at the ACA. You end up with this lariat structure where you have a 2' to 5' phosphodiester, and these guys get ligated together. You have nuclear export and translation. What happens to this? This is what we haven't talked about yet. So this lariat, the first thing that happens is it gets broken down back into a linear RNA. And what does that is this protein called the debranching protein, Dbr, which will break this 2' to 5' phosphodiester, and then this intron can get chewed up and recycled all the nucleotides, but also there are quite a few introns that encode SNO RNAs, which we'll talk about in just a second, and microRNAs that we'll talk about at the end of the term. So actually there are a number of RNAs that are encoded in introns that turn out to be very important for regulatory purposes or for modification purposes. But most of the time, this now linear RNA is going to get chewed up. Now, why does it get chewed up? This 5' end, what does it have on it? Phosphate. What does this guy have on it? Your messenger RNA? It's got a cap. So this is going to be a substrate for degradation. What's usually present at the 3' end of your messenger RNAs? Poly A tails, protecting them for degradation. Does this guy have anything that's protecting it from degradation? So usually this is gotten rid of really, really quickly. And so this gets us to the surprise of the next paper, which was here, that in stationary phase, this group saw that there were lots of introns still remaining. And so how did they do this? They used a technique called Rna sequencing, or Rna-seq is what everybody calls it. So we mentioned genomic sequencing. We've talked about some of the techniques, the Illumina sequencing, et cetera, et cetera. With a lot of these sequencing techniques, you get very short sequences. And so basically what was done here is they purified all of the RNA from their yeast cells and made DNA copies of it and sequenced a whole bunch of them. And then what they did is they took all of these short sequences and lined them up relative to their genome sequence. And so that's all that this is right here, is that you have a sequence relative to the genome at the bottom. Every time you have a sequence, you will start to make a little bit of this histogram going up here. So in this case, this is the actin gene. You sequence all the RNA. You have a bunch of RNAs that are here, very few RNAs that are here, a bunch of RNAs that are here. What does that mean about this sequence here? Because it's the RNAs that you've isolated from the cell. Isolate the RNAs, and you have a DNA copy of it, you sequence all of it. What does that mean about that particular part of your sequence? It's an intron, exactly. So it's gotten chopped up and is no longer present in the RNAs that you've isolated. It's both from log phase cultures and saturated cultures. This was true for most genes, but there were a few, which was very strange, where they saw in saturated cultures lots of sequences that corresponded to this region right here. Same thing true down here. Here's a gene. In log phase cultures, there's nothing there. In saturated cultures, you could also call what you see in stationary phase a bunch of sequences. Very strange. Now you've got all these introns that are present in stationary phase. Now, if you see something like this with an RNA-seq experiment, you really want to confirm that's what you've seen. So how do you confirm that you have a particular RNA which is being made? You do a northern blot. So in this case, the northern blot is all of the RNAs under a particular condition. So this is log phase growth or stationary phase growth that you now hybridize to a complementary sequence to the intron. So in this case, your complementary sequence to the intron right here only is present in stationary phase, not in log phase. And that actually parallels really nicely this. The sequences, you found all these RNAs which corresponded to the intron, and you can see that particular RNA. Down the bottom here is something which is really, really important. 5.8S ribosomal RNA. So what would that be? The same amount, more or less, between your log phase and your stationary phase. What this is saying is the absolute amount of RNA that you put on your gel is the same for each of these. So this is your control that says, I just haven't put a whole bunch more RNA here, and that's the reason that I detect this, that you've got the same amount of this non-related RNA means that this is specific then to stationary phase. Same thing is down here for this SAC6. So that's great. It's wonderful. Look, we've got these introns which are still present in stationary phase, but they're not present in log phase. And this group took it one step further. Again, this is yeast, so you've got a whole bunch of interesting mutants. This is the Dbr1 mutant. What does Dbr1 do? It debranches introns, so it takes those loop structures and linearizes them. Well, if you don't have that particular gene, you're not going to be linearizing your introns, so you actually end up with these really big pieces on northern blots, which are just stuck, and they don't get through the gel anywhere as easily as these linear pieces. So it's a debranched part here, so they undergo debranching, but they're still present in stationary phase. They're not present in log phase, but they're present in stationary phase. So what do we think is going on here? Now this is hand waving. There's a lot more experiments. If you'd like, you can go and check out these papers where they look at what they think is happening and why, in the first case, when you do deletions of introns, cells have trouble in stationary phase. And in the other case, in stationary phase, you have these introns which are being maintained. And the basic story seems to be is that these introns are actually keeping splicing proteins away from splicing other things. And those other things turn out to be ribosomal genes. And so ribosomal genes, these are what you need, and these are the ribosomal protein genes because they're the ones that have the introns in them. Ribosomal protein genes really control the growth rate and literally how much energy the cell is spending. So if you think about stationary phase, you want to conserve energy in that stationary phase. So conserving energy, what's the best way to conserve energy is have fewer ribosomes. Spend a lot less of your energy doing translation. And if you don't have your properly spliced ribosomal genes, then those are not going to be made into proteins. You're not going to be making them into ribosomes. You're not going to be spending as much energy, and through that whole process, you end up surviving better. So introns in this case actually seem to be a way of helping cells to survive under starvation conditions. Just really kind of cool. So it's not just, introns aren't just for alternative splicing. They also seem to be involved in helping out with stationary phase. So I thought this was really cool, so I wanted to introduce it here as well. So we've done capping, we talked about that last time. We've done splicing that we talked about last time, and today what's left? Poly-A tailing. So 3' end of our messenger RNAs. How do we get those? So the way that you get poly-A tails These are signals that are present in the messenger RNA as it's being transcribed. As the messenger RNA is being transcribed, it'll come across some certain sequences. No, I do not expect you to remember these sequences. I can't remember them either. And then you hit a particular sequence, which is also after it's been transcribed. So this whole thing has been transcribed. All of this is in your primary transcript. Then this primary transcript gets cut. Once it gets cut, you have a 3' OH. Poly A tails get added, and you have your final messenger RNA. This, which actually is still being transcribed, there's an RNA polymerase sitting over here, transcribing its way along. This gets chewed up, and actually it turns out the chewing up of this is probably very important for transcriptional termination. So transcriptional termination in eukaryotes is a little different than what we have in bacteria. But let's first talk about making the messenger RNA. Again, first thing that happens, you have to have this cleavage event that takes place. So how do you get cleavage? You have a sequence in your messenger RNA that binds to CPSF. And yes, it's in red because I do expect you to remember this. Cleavage and polyadenylation specificity factor. So cleavage and polyadenylation specificity factor, if you remember what it stands for, it should be pretty obvious, gives you this specificity where you're going to cut your messenger RNA in order to make a poly A tail. That's not the only factor you need. You also need a cleavage stimulation factor, CSTF. Both of these are associated with the CTD, the C-terminal domain of RNA polymerase II. So they're brought along as the polymerase is transcribing, as the RNA is being made. Once this particular sequence is made, CPSF binds, gets CSTF, it's cut. There's lots of other factors that we're not going to talk about here, so I haven't put them in red. Then you have the poly A polymerase. Poly A polymerase does what it says, takes adenines and adds them in a template-independent manner. So this is actually the first sort of polymerase of any kind that we've talked about has a template-independent way of adding nucleotides. So literally just adds A's, and usually on the order of a couple of hundred of them. Once you have a couple of hundred A residues, you have binding by the poly A binding protein, PABP. This should be all of the acronyms that we're going to talk about as far as termination is concerned. So you have the poly A tail, which is formed, poly A binding protein, and this is now your final messenger RNA that can get exported from the cell. But the question is, what happens to the RNA here after you have cleavage that takes place? And so that gets us to transcriptional termination. You have the poly A tail, which is added here after the binding of CSTF and cleavage and polyadenylation specificity factor. And now you have an uncapped end of your RNA. And so this should be sort of a recurring theme. Why do you need caps? So they don't get chewed in at the ends. If you don't have caps, what happens? They get chewed in at the ends. So what happens here is you have a nuclease that has a name, but we're not going to remember it, which chews in at the 5' end. So it's a 5' to 3' exonuclease, which literally just chews up the RNA until it catches up with the polymerase. And then this piece of RNA is actually too short to associate with the rest of the RNA polymerase. The RNA polymerase falls off of the DNA, and you start everything again. So it's literally just getting the RNA out of the RNA polymerase. And just as a reminder, how you get transcriptional termination in bacteria, you have this secondary structure and a U structure. Somebody asked me after class, yes, there's another kind of way you can get transcription termination in bacteria. Also has a protein. That protein actually pulls the RNA out. But it's all about getting the RNA out of the RNA polymerase. Once you get that RNA out of the RNA polymerase, transcription terminates. The RNA polymerase falls off. So in the case of bacteria, you have this secondary structure and the U-stretch, which allows that RNA to basically pull itself out of the RNA polymerase. In eukaryotes, you have degradation of this free end, which is generated when you have poly-A tail formation, and then that pulls off the RNA, and then, again, different mechanisms in bacteria. There's another protein that literally pulls everything out. So that's termination and poly-A tailing. Turns out that all of these things are happening simultaneously, and this is something I mentioned last time as well. How do we get everything happening simultaneously? It's because all of the factors that you need, and factors being protein, and in the case of the spliceosomes, RNA, which will be processing the messenger RNA as it's being made by the RNA polymerase II, because all of these factors are associated with the RNA polymerase together with this CTD, supposed to be shown as phosphorylated here with all of these extra little balls coming off on the side of it here. And just one example, I don't want to show you the data, but it's been really clearly shown that the cleavage and polyadenylation specificity factor actually interacts directly with U2, SNRNP. So you have interactions between the cleavage and polyadenylation factors and the spliceosome. We also know that there are interactions between spliceosomal proteins and capping proteins. So all of this is really happening simultaneously. We talk about 5' end splicing, 3' end. It's happening as you're getting transcription taking place, and there are lots and lots of proteins that are involved in this process. And so we always cartoon these as being nice separate RNA polymerases, separate proteins relative to each other, but they're all really in one kind of giant god-awful mess. So how does this all get organized? And this is what people call the messenger RNA factory. If you look at nuclei, and in these particular nuclei, and just stain for these different proteins, so the RNA polymerase, the capping enzymes, the splicing enzymes, etc., all of these localize in specific places in the nucleus. They're not all spread out. And so if you did have all of these RNA polymerases, cruising along a particular gene on a particular chromosome at a particular time, you'd expect this to be really fuzzy because all of these proteins would be in different places in the nucleus at the same time. But they're not. They're actually all concentrated in one place. And so this gets back to what we talked about before, is that it's probably the DNA which is being moved to some of these areas where all of these proteins are associated in order to make the messenger RNAs. So it's the bits of DNA that are being moved to your transcription factory or your mRNA factory in order to get transcription to take place. So, and this also makes sense when you think about the whole genome. A lot of the genome is not transcribed, at least not being transcribed into messenger RNA. So you don't want to have all of that genome in where you've got all of the proteins that are going to be doing all the jobs that you need them to do to make the messenger RNAs. So now we've made our messenger RNAs in these RNA factories. What do we need to do with them? We need to get them outside the nucleus so they can actually finally be translated. How does that work? Well, literally, if you have a really good electron microscope, you can see the RNA as it's being made and getting modified and gets stuffed out through the nuclear pore. Well, that's what you can see. That's about the highest resolution you can get with an electron microscope. But if we now look at what proteins are associated, we can do the cartoon form that we have down here. So one thing about having all of this messenger RNA modification machinery, capping, tailing, splicing, etc., you want to make sure, to totally over-anthropomorphize here, that these messenger RNAs have been properly processed so they can get translated, and that they get translated properly. You don't want to have an incorrectly formed messenger RNA sent out into the cytoplasm. And this also gets back to what we were talking about, some of the uses of potentially of introns, is you don't want to translate things that you don't want to translate. So really saving energy. Any kind of problem that you've had with your messenger RNA, you don't want it to be translated, because again, that's the big energy consuming step. So how does the cell know, quote unquote, that all the appropriate processes have taken place? Well, we've got our cap binding complex that binds to a cap and only binds to a cap. So if you have a messenger RNA that doesn't have a cap on it, it's incorrect, or it's certainly not a messenger RNA. So cap binding complex is something that you need. We talked about what are bound to introns. You've got lots of these heterogeneous nuclear RNPs. Most of these have to be no longer on your RNA. One of the things that we didn't talk about is after you have splicing, there's this thing called the Exon Junction complex, EJC, which binds to the 5' ends and 3' ends of exons and actually overlaps between them. And it's only associated with messenger RNA after you have splicing. And so it's another way of controlling that splicing has actually taken place. What about the poly A tail? How do you know if you got a poly A tail? Structure, but what else? What binds to the poly A tail? Exactly, the incredibly creatively named poly A binding protein. So it's all about the proteins here. It's not about the RNA. So it's all the proteins that are being recognized. Proteins associated with the RNA, and it's that complex where you have your cap binding protein, your exon junction complex, and poly A binding proteins. SR proteins are often associated with this. You remember, SR proteins are sitting on exons, and so you know that you've got them on the exons. Some of them are maintained, some of them are not. And then got all these proteins together, you get the right constellation of proteins. This will then get exported out of the nucleus. How do you get something this big through a nuclear pore complex? You have to have nuclear export proteins. So this will bind to this complex of proteins in RNA, get it out of the nucleus, and then this protein is removed, goes back into the nucleus. The cap binding protein is actually removed and goes back into the nucleus, and then you can start to do translation. So export is really about quality control. You have caps, your splicing has taken place, and you have your poly A tails that have formed. And if you have that appropriate combination on your RNA, then and only then can it get exported. Yeah, John?
Sounds like all of that list is like sort of a consensus. You have to have all those things. Where would that take place? It would have to take place before the pore in order to save work.
Yeah, so the question is, where is this happening? And to some extent, where is this happening relative to what we just talked about, where you have your messenger RNA factory, where it's making all of these things in the first place? And so one of the things, maybe we should back up here quickly. You may notice that a lot of these things here, all these spots, a little hard to tell, are actually pretty close to the outside of the nucleus. So they're relatively close to the nuclear pore complex in many cases, so it's not too far to go. But definitely getting from that messenger RNA formation, been released now from the RNA polymerase, so it's certainly no longer directly associated due to that. But then these other enzymes, so basically once they've done their jobs, then it can move. But then there's the quality control of these, oh, sorry, wrong way, nuclear export proteins. And these guys sit right next to the nuclear pore. And so this is only going to associate with your RNA once it has all of the modifications that have taken place. And again, modifications because of the proteins that are now associated with that RNA. So it seems that you get all of the processing then you have the quality control of your nuclear export protein, and then it can get exported. But it's all happening in the nucleus before you get out. What happens to the rest of the RNA? It gets chewed up by the cellular RNA garbage disposal, I like to call it, or the exosome. They just love these zomes. But the exosome is literally just a whole bunch of RNA exonucleases. And so what it does is it just chews up any RNA that doesn't have a cap on it and doesn't have a poly A tail to protect the end of your RNA. This again is oversimplification. It always is. A lot of the smaller RNAs that kind of mentioned snow RNAs and microRNAs that sometimes come out of introns, those usually have pretty strong secondary structures. And so it turns out that it's literally the linear form of RNAs which get chewed up. And the reason that that happens is if you look here, all of the actual chewing activity takes place right in the middle here. And the only thing that can fit into here is going to be a single-stranded RNA. So a single-stranded RNA will get in there and then get completely chewed up. Okay, questions on messenger RNA processing? More questions on messenger RNA processing. Then I have a question on messenger RNA processing. Termination of transcription by RNA polymerase II in eukaryotes is due to which of the following? Exonuclease activity, endonuclease activity, lots of U's in the transcript, hairpin structure in the transcript, or C and D. And I should start it, shouldn't I? That would help. Ten. Okay, we still don't have a consensus, so let's see if we can get one. Okay, we ready to go again? Go. Again, you can continue to discuss what you thought or not, as the case may be. 10, 5, so it's definitely not C, D, or E because that's prokaryotic, and actually I should have put bacterial transcription termination. So you end up with exo or endo, and you could probably argue a little bit that endo might possibly be right because you have to get endonuclease cleavage to get a 5' end before the exonuclease can chew in and really displace the RNA. But it's really about getting that RNA out of the RNA polymerase. So it's an exonuclease, which gives us the termination of transcription, again, in eukaryotic cells. So just finish up, and we're probably not going to get to translation today, which is perfect because it's not in the syllabus. It's supposed to be on the midterm anyway. So just talk a little bit about ribosomal RNA processing. So far, we've only talked about messenger RNA processing. Ribosomal RNA, it's the most common RNA that you find in any given cell at any given time. And in eukaryotic cells, it's made by a separate RNA polymerase, RNA polymerase 1. RNA polymerase 1, it's got a couple of differences from RNA polymerase 2, but probably the most important as far as modifications are concerned, it doesn't have this C-terminal domain. So it doesn't have all of those extra modification enzymes that's associated with it, which is probably why the ribosomal RNAs don't get modified. They don't get caps put on them. They don't get spliced. They don't get tails put on them. But they still have to end up being pretty heavily modified because the ribosomal RNA transcript is actually a precursor RNA that gets chopped into smaller pieces and heavily chemically modified. And this is a chemical modification that happens to the RNA. So in messenger RNAs, there's not very much chemical modification. We'll talk about some later because there is. But at least as far as we've talked about so far, there's no chemical modification to the RNA itself other than that extra cap that gets put on at the 5' end. So on the other hand, ribosomal RNA has lots of chemical modifications that take place to the RNA itself. And it turns out the chemical modifications to the RNA itself are absolutely critical for the activity of the ribosome. If you don't have some of these modifications, the ribosome is not active, and we've mentioned this before, but I'll mention it again a lot more when we talk about translation after the midterm, that translation is really catalyzed by the RNA part of the ribosome, not the protein parts of it. So how do we go from this primary transcript to the different RNAs that end up getting put together into the ribosome? So you have cleavage that takes place and chopping of the ends here. I'm sure all of you remember the number that we skipped when we talked about the small nuclear RNAs. We had U1, U2, right, so U3, U4, U5, and U6. U3 is actually involved in some of these cleavage steps in chopping apart these individual pieces. But then how do you get these modifications in the ribosomal RNA? The way this happens is through the SNO RNAs, not to be confused with the SN RNAs. So these are small nucleolar RNAs. A small nucleolar RNA plus a protein is a SNO RNP. So just like we had with the spliceosome, SN RNAs, SN RNPs, now SNO RNAs and SNO RNPs. Why SNO? The NO is for the nucleolus, separate-ish compartment inside the nucleus. You can also think of this as kind of the ribosome factory, where all of the ribosomes get their modifications that take place. So what do these SNO RNAs do? The chemical modifications that happen to ribosomal RNA have to happen at very specific positions in the ribosomal RNA. So how does the cell know what position to modify? The way it knows the position to modify is through base pairing interactions. So the base pairing interactions that happen between these SNO RNAs and the ribosomal RNA tells the modification machinery that's where you need to make the modifications in the ribosomal RNA. The two most common of these modifications are pseudouridine and 2'-O-methyl bases of any particular kind. Pseudouridine is just literally taking uridine and moving it around. So instead of where you have your normal uracil, this has just been isomerized. O-methylated nucleotides, we've already talked about this at the 2' position. You end up putting a methyl group on them. So again, these have to happen in very specific positions all throughout the ribosomal RNA, and lots of the different ones, the large ribosomal subunit, the small ribosomal subunit, etc. How do these happen at the specific positions? There is a SNO RNA that will bind to a specific position in all of these places in your ribosomal RNA and cause the modification to happen at that position. So you actually have to have, for every one of the modifications that take place, so this circle here or this triangle here, you have to have a complementary SNO RNA to that sequence. Now there are two major groups of these SNO RNAs, the CD box and the HACA SNO RNAs. These just have conserved sequences that are present in these RNAs that are not the sequences that are complementary to where you're causing the change. So these conserved sequences, these are the conserved sequences that interact with the proteins that will give you this modification. So the box HACA, say box HACA SNO RNAs, here's your hairpin. These are important for pseudouridine. The CD box SNO RNAs, that CD box. Remember the Tata box from last time? What's a box? It's just a conserved sequence. So CD boxes, all of these SNO RNAs have conserved sequences that are associated with them. That's going to interact with this methylase, which will methylate the riboses. So in the nucleolus, that's where you have all of these modifications taking place. All of the transcription, actually, the ribosomal RNAs are found in the nucleolus. The RNA polymerase 1 is in the nucleolus. It's making these RNA genes. And actually, I should have mentioned, the RNA polymerase 1 just by itself is insufficient to make enough copies of this ribosomal RNAs that you need to make all the ribosomes. Again, 80% of the cell is ribosomal RNA. Or I say 80% of the RNA in the cell is ribosomal RNA. So you actually have many, many copies of the ribosomal RNA genes, usually a couple of hundred copies of the gene. And so that's what each of these is supposed to represent up here. Each of these are many copies of absolutely identical sequence here. So that gets transcribed. This then gets chewed up by U3. The SNO RNAs will bind specifically with the parts of the SNO RNA that's antisense. The conserved parts will bind to the proteins, which are going to do the modifications. Once you have all of these modified RNAs, then you add all of the proteins to these ribosomal subunits. Then all of these ribosomal subunits eventually get exported out of the nucleus, and you end up with functional ribosomes out in the cytoplasm. Turns out it's not just ribosomes which are made here. tRNAs are also made in the nucleolus. Some of the small nuclear RNAs are made in the nucleolus. And some people call this the Ribonucleoprotein Factory. So putting all the proteins and the RNAs together to get all the RNPs that you need to finally make all of your messenger RNAs. So you have messenger RNAs. If you're smart, you do the prokaryotic route. You don't bother with all of this extra stuff. You transcribe and translate it directly. However, if you're a eukaryote like us, you've got lots of exons and introns. The cap gets made at the 5' end, so you've got the cap binding complex, and you protect it from the hungry exonucleases. Introns get cut out, exons get put together. The polyadenylation happens because you have your CTSF and CPSF binding here, poly-A polymerase. All of these modifications are recognized through protein interactions with those modifications, and those proteins together with the messenger RNA are exported into the cytoplasm where they get translated, which we will talk about after the midterm. So review on Wednesday and midterm on Friday. Bring your scantrons. If you're not taking the exam here, let me know, and we'll figure it out.